my dissertation presentation and defense. Today, obviously you can see that my dissertation is entitled Comparative Topological Analysis of Neuronal Arbors via Sequence Representation and Alignment, and hopefully by the end of this presentation that will all make sense. And my committee members, my committee chair is Dr. Giorgio Ascoli, and my other committee members are Dr. James Olds and Dr. Joseph Weissman. There we go. So I'll start off with an outline here, which actually corresponds to the first chapter of the dissertation. I'll start with motivation and background, dealing with neuronal morphology, what that is, why we're interested in it, as well as a bit about neuronal reconstructions, which are at the heart of analysis of the data. And then I'll follow up with dealing with that morphological reconstruction process, which is a major part of the field producing the data, and the diatom challenge, which was an effort that we undertook to really move that part of the field forward. After that, I'll get into really the heart of the concept that I've introduced, which is presenting neurons as sequences, which then allows us to do various types of sequence analysis. And I'll start off with a motif analysis, which is looking at short subsequences, and then get into a blast-like global alignment and topological feature identification. So neurons have a really broad diversity in terms of their morphology, as well as other features, and this comes from the various different functions they have, from specialized sensory neurons, motor neurons, and everything in between in the neocortex, pyramidal cell, which are sort of the primary information gatherers and passers, as well as various types of inhibitory interneurons, which are involved in synchronizing parts of the brain, as well as distinguishing certain ideas or perceptions from others, and so on. And so here's a pyramidal cell, as opposed to this is a Purkinje cell from the cerebellum, and I believe this is a granule cell. There's a whole bunch of others, and you'll see a selection. And we measure them in a number of different ways. So certainly there are features like diameter and the taper of branches, as well as the tortuosity, how they curve through space, and the angles of the bifurcation. And so these are all branch level features that can be useful in understanding what's going on with a particular neuron or particular class. And then we can also look at features like the whole size. How much space does the neuron take up? What are its dimensions? How much total length does it have? And so all of these can tell us quite a bit, and we can use those to correlate with various other functional properties. So one of the major functional properties that we a lot of times will look at is the firing patterns. So you can see here with various different morphologies, you have very different patterns in terms of how fast the frequency, high the frequency or slow the frequency is, as well as other bursting type patterns. So there's quite a broad diversity of the way that a neuron can act electrophysiologically. You can also look at things like the topology in terms of the branching structure, and that is impacted by a number of different processes. This particular case shows a distinction between minimizing the total amount of wiring, which is really important for conserving energy, as well as reducing the amount of volume taken up by a particular axon or dendrite. And that can be balanced against the amount of time it takes for a signal to propagate from one part of the neuron to another. And so if you balance those, you get something much more realistic as opposed to a case where you're just minimizing wiring. And so this balance is also something that we can look at and look to morphology to help us understand. One of the ways that we measure topology is by looking at asymmetry. So asymmetry, in terms of the technical measure, is actually the average of partition asymmetry across all of the branches. So if we look at just a single schematic here, right, this is completely symmetric, so it has an asymmetry of 0, and this is completely asymmetric, so it has an partition asymmetry of 1. And we can look at that in terms of the number of branches, the length, and other features as well. And so this has been a very useful statistic throughout studies of morphology. We can also look at something more specific, right? So this weights evenly, and that does not provide the full discriminative ability in terms of topology. So one thing that Giorgio Ascoli, Carrie Brown, and I developed a few years ago was a measure called coalescence, which is based upon this partition asymmetry, but looks for the main path. And so here, it's a bit hard to see, but this yellow path is the main path of this apical dendrite, and there's a number of main paths coming off of the basal dendrite of this pyramidal cell. But this is really, we're looking at the prominence. So if we actually take a measure, this is going to be much stronger in terms of its prominence than these ones. However, as useful as that can be for some cases, we can also see a case like this where it's a bit hard to say. We can actually get a measure for this whole neuron, but the problem is that in certain places it looks like, well, there might be high coalescence here, but if you take this whole tree, it might be low, right? So that gets into the question of, are there perhaps more finer features that we can detect and use to measure? So one of the nice things about looking at topology is they're least dependent among many other features that we could measure on imaging method and noise, the format that we use to store the data, and the experimental method used. And all of these different things can have an impact on the diameter being one of the things that's most challenging to be in terms of its consistency. And so topology is relatively less impacted. We can also then look at topology to figure out things like, what are the mechanisms underlying growth? What are the metabolic and volumetric constraints, as I mentioned earlier? And also, there's certainly an anatomical effect in terms of the particular region that any given neuron is a part of. And we can look to these patterns perhaps to tell us something about signal processing, as well as the connectivity and spatial distribution, right? And that's really a requisite part of how we get the connectivity in the brain, which allows us to think the particular thoughts or sense the particular senses that we have in the way that we do. Right? And so here's just two rough schematic examples of sort of opposites in terms. So here we have a high asymmetry at the top and a low asymmetry here, and the opposite. And so we're looking for patterns like this, but really we could look for any patterns, and really the question here was to do an exploratory search and not necessarily to say ahead of time what we expected to see. So another part of this process I mentioned before is the reconstructions. And we use neuromorpho.org for the data. This is a collection of reconstructions produced from various labs across the world on a slew of different species, brain regions, cell types, and so on. And this is really important, this curation process, in order to actually know what we're looking at and get the right data and exclude the data that's not going to be relevant for the particular analysis. The other part of this is actually producing that data. So there's a lot of data that's been produced in the past, but we're looking towards the future in which we're really expecting high throughput, right? So we've heard of high throughput genetic analysis. We'd like to be able to do that with neurons and their morphologies. So there's been a lot of work recently in terms of imaging as well as reconstruction processes. And this is the process of reconstruction, where you have an image from a microscope, assemble multiple images together, and then take these and trace paths. It's a bit hard to see here, but ultimately you get a digitized version. And that is great in terms of it reduces the amount of storage, and it also is very amenable to all kinds of different analyses. And so this is the sort of format that we used. So in that process, there was a question a number of years ago of where are we really in terms of the state of automated reconstruction, right? Because that's ultimately the future. It can take weeks to months, depending upon the neuron that people are reconstructing, to actually go from a microscopic image to one of these digital files, depending on the size and various other factors, right? So we wanted to know what the current state was, and there was certainly a little bit of a disagreement between the neuroscience community and the computer science communities. So we had an opportunity to actually test this. And the other main question, of course, is what are the major bottlenecks? Because if we know that, we can really make some more significant steps forward. So we held what was called the Diadem Challenge, the digital reconstruction of axonal and digital morphology, had a competition, had a conference, and found out quite a fair deal, and that's really helped push the field forward. We have a website that can be seen that has a recounting of all of that process. But in that process, one of the big questions was, how do we quantify quality? Right? We want to see actually how good an automated reconstruction is. If we can't tell that, then we really can't know if we can use a particular algorithm or its results. So one of the things that we looked at was the judgment of data users. So there are scientists that do analysis, and we can also look at judgment of the data producers, right? Sometimes those are the same people, but certainly there are other people that use the data other than just the individuals that generate it. And then one of the other thoughts was, well, really what we want to do is minimize the amount of time that individuals have to take to produce this data, right? So how much time is saved for them? And then ultimately, because we want to have speed in terms of doing this quality check, and we also want to try something that's unbiased, right, not subjective, I created an algorithm. This was, again, with Georgia Rascally and Carrie Brown, another project we worked on together. And so we ended up calling that the diadem metric. And so, we're looking at, these look very similar, but they're a little bit different, right? Can we detect those differences and produce some sort of a score? Here's some actual examples where we have a gold standard. This is a manually generated, expert reconstructed file, as opposed to 3 different automated reconstructions. And you can see right off that there's some differences. And so we need to be able to determine, right, how close to the true, right, ground truth or gold standard are those. So as I said, we compare to a gold standard manual reconstruction. The scoring is something that there was a lot of question about. We decided to score based upon the functional effects, the topology, right? So we weighted by the branch importance. How big is the subtree coming from a given branch? And we detected correct and incorrect branches. So here, this is a gold standard. This is an automated reconstruction. And you can see just by eye, it's pretty difficult to actually tell if the connectivity is correct or not, right? So you really need to zoom in and take a lot of time to look at that, whereas an algorithm can make those detections very quickly. And even things like this, where you have a point that's correct, it's not connected correctly. So what we found was that this data is at least as good as expert judgment. So this is one different type of score plotted against another type of score for the same reconstruction. And it's a little bit difficult to see, but you can certainly see that there is a correlation here, which we would expect, right? We want the scores to line up for the different methods, but it's not perfectly correlated. And when we compared the qualitative score of expert judges as well as the interaction time score to the metric, we found actually an even better correlation than those other types of scoring to each other. So it showed that we had something that was definitely useful, and indeed, it's been used since in 7 publications and cited in 29, at least as far as I'm aware of. There may be more by now. And it's also available open source. Individuals can use it just for checking as well as for getting feedback on where their errors are, and they can modify the scoring parameters and such. So now once we have the data, I'd like to do something to figure out more about why neurons behave the way they do and what drives them in terms of their morphology. So the idea that I believe Duncan Donahue and George Rascally had some years ago was, what if we represent neurons as sequences? Can we then leverage some concepts from genomics, right? Because they dealt with big data, right? This large data sets that are really beyond anything that a lot of previous algorithms were able to handle, and still detect important relationships within that data, right? But also the nice thing with this is we can avoid, or another thing that we wanted to do, excuse me, was to avoid relying on arbitrary metrics. So one thing that you can do is do feature selection, right? And so that's a good option that many have and use in their analysis. But we can also do something where we don't rely on that at all. And we can just sort of let the features emerge from the process. So the process that we start with is, well, we need to actually turn every bifurcation into a character so that we can make a sequence. First thing we do is break this down by by particular bifurcation types. And so just to make clear here, we're using binary trees. That is, trees that every time there's a branch, there's 2 children coming out of it. Real neurons do oftentimes, or occasionally, have trifurcations or more branches coming out of any given node, bifurcation node, or excuse me, branching node. But it turns out it's at most 3%. I did an analysis on the neuromorpho.org data and found that that's about the max it was. For several of the particular arbor types, and I'll discuss the differences there, it was actually more like around 1% or less. So the conversion to bifurcations is a pretty simple process, and in the vast majority of circumstances should not have any significant effect on the results. And so here, just also for, you'll see this a number of times, this symbol here is just to show that there's a bifurcation or a whole subtree of indeterminate size as opposed to a termination. So we have an A, a C, and a T. These are, yes, related to the nucleotide sequences, which is one of the underlying ideas. And in this case, we're calling this an arborizing bifurcation in that it adds a new subtree to the tree. A continuation bifurcation, which doesn't add or remove because one side terminates while the other side branches, and a termination bifurcation because both of its subtrees terminate. So the process then, we have a number of different options. There have been a lot of good ideas, but we decided to go with a depth-first traversal, which means that we go all the way down a full subtree and then come up and take the next subtree. So this process, in this case, we're going down a smaller subtree first. So we'll go down here and then jump back up and so on. And so here, just sort of show you the process on an actual neuron and how this goes. So at this point, we'll come all the way back up to the initial root again and go down its other subtree. So one of the things about going down the smaller subtree first that's nice is it keeps the nodes. So for instance, this node is closer in the sequence to its parent than it would have been than this node would be if we went down this subtree first. Right? So we're keeping as much as possible the child nodes of any given parent node close in the sequence. There are other methods. For instance, you could have a different sequence for every single path, and that's actually been used in other analyses, but we decided to keep one sequence which is as long as possible to really look for the patterns that we're interested in. And so here's an example of how we actually broke these up. So we know that axons and dendrites are very different in terms of what they do in the neuron. So we broke those up into separate analyses. We also look for pyramidal cells, which I had mentioned before. They have a very clear separation between what's called the basal arbor and the apical arbor. So we also split those up. And apical dendritic trees are combined together into a single sequence. So this is a representation of this apical arbor, and here's the representation of the basal arbor all together, and here's its axon. You can see how big the axon is in relative terms. Now, it is also the case that not all axons, because of how big they are, actually most of them are not fully reconstructed. So that's something that is in the analysis that we do try to adapt for, and I'll explain how in a bit, but it also has to be kept in mind in terms of whether the analysis is reflective of the true morphology or perhaps if there's this artifact at play. So one thing that we did, because we're really looking at topological patterns which involve complexity, we stuck with arbors with at least 20 bifurcations. If we went too small, then we basically would have a lot of neurons, the smaller ones, that have the same topology, right? Just by chance, because there's just not as many shapes or separate topologies. In terms of species, this is what was in the data set after getting rid of non-control cases and some other data that didn't quite fit with what the sort of analysis we wanted to do. So we've got mostly human, a lot of mouse and rat. In the axons, rat, mouse, and we've got some Drosophila. And in the apical dendrites, rat, mouse, and monkey. So obviously there's a lot of rodent in the data set, and the various different sub-data sets, a few different other species come into play. So we get a nice chance to compare different species that way. In terms of brain regions, both dendrites and axons are mostly from the neocortex. We've also got some hippocampus and retina, as well as for the axons, hippocampus and Drosophila olfactory bulb. So we do have some sensory as well as cortical neurons to compare. And in terms of apical dendrites, most of them, right, most of the pyramidal cells that we have are from the neocortex, and some are from two different regions, the CA1 and the CA3 regions of the hippocampus. And that's actually important because those are certainly known to be different, and in fact in the results we do see the differences between them. So they're all pyramidal cells, but those apical dendrites differ between those regions. So the first thing we start with is motif analysis. We're looking for short subsequences that are different between different neuron types. And so one of the things that we can look at is the length and the proportion of one node type in terms of just these node types, because the number of those T nodes is equal to the number of the A nodes plus 1. So basically we've got two equations. We've got 4 variables, and that means we need two measures to capture all of the information. So what we end up doing is we look at the length and the proportion of those C nodes. And so here's just a quick look at some neurons in the percent C. It's a little bit difficult to see, but the point here is really just you can look at these and you can see certain features, but seeing the topology in one of these reconstructions is definitely very tricky by eye. So this analysis is really useful to pull that information out. So here we see the percent C in dendrites and axons, and they're very different. There's definitely a fair bit of overlap, but we can see a difference here. And when we look at the pyramidal cells, the dendrites, basal dendrites and the axons actually are pretty reflective of the overall data set. So there's not a very significant difference there that we specifically look at. But the apical dendrites fall really in the middle, right? So that's already the first indication of differences between basal dendrites and apical dendrites. We'll see plenty more. Another thing that we notice, though, is that these dendrites actually, as has been reported for subsets, certain sort of more restricted set of cell classes, that they correspond with this terminal growth. So we have different ways that we could grow a neuron, right? Of course, they grow in whatever way they grow in, but we can have models. And so one of the models is simply to set a size and grow it. Okay. So in this case, if we started off with this tree, we could grow at any of these tips for terminal growth. And this represents the distribution of that process, which corresponds nicely for the dendrites. For the axons, they're a lot closer to the segmental growth, although not quite to that same level. And so that's basically any branch can grow in the process. But if we look at the sequence length, which I had said before, we really need both the proportion of C nodes and the sequence length to know what these neurons are made up of, what their, that part of their structure is. And it turns out that for dendrites, it actually shoots up as once they get greater than 40 bifurcations and starts to get very close to the axon level, which doesn't really change too much. So it looks like perhaps larger dendrites are larger because they have more of these side branches, these branches where one of the children terminate. But you can see also most of the analysis for the dendrites is really going to be in this region because this is the amount of data. Right? So there's a really large amount of our data is in this size. And obviously there's a lot more over here that we cut off because we weren't interested in those that were too short. Axons, though, have a much wider distribution. So one of the things we also wanted to look at was that increase in C, does that have anything to do with where the C nodes appear, these side branches? Do they tend to happen further out in the dendrites, perhaps, and that's why with the larger ones we see that go up? We looked at that and we saw over length, right, this is the relative position within a sequence, that here's the proportion of C nodes, right, which is close to that original 30%, and it goes up for the larger dendrites, but that distribution across the position, across the sequence does not change. Right? And in fact, the other nice thing is we get to see that there is an effect of the terminal growth, sort of a signal of that in all of those cases. That's this rise, early initial low percent C with a rise. Right? So basically you can imagine in neurons that at the top of neurons, sort of near the root, there's a lot of branching going on and not a lot of terminating. Right? And then later on, you get more of a balance between those cases. So now if we want to look at larger patterns, right, we weren't just interested in node types. Some of that's been done before, not at the scale or the amount of neurons that we looked at, but we wanted to see what about sequences. So we're looking at a subsequence of length k, we're calling a k-mer. We're then going to say those that are overrepresented are called motifs, those that are underrepresented are called anti-motifs. So this is, the way that we're going to approach this is more of a graph theoretic approach as opposed to a genomic approach for those who could potentially be thinking about the genomic motifs. For those not, don't worry because I'll just focus on the one that we're doing. So to start with, these are dimers, right, 2 mers. And here there's 9 of them, 9 possible ones. Here's just some examples in an actual tree. And one thing that is worth noting is there are cases, because this is a tree structure built into a sequence, there are special cases that come out. So for instance here, we know since we're always going to the smaller side first, if you have a T followed by a T, you can't have anything other than an A before them, right? Because if you had a C here, that would make the first subtree larger, right? So that can't happen. So for instance, CTT is a trimer that doesn't exist in this method. But we also know that when we have a TT, it is an A. And so this also shows the number of k-mers that we have when we increase in K. So it goes up quite a bit. And that leads to the question, well, at what point do we want to stop? Because we're going to get a lot of k-mers up here. And there's also the possibility that we may not really see any important information at a certain point. So we'll address that in a moment as to how we figure out where to stop. But first we need to actually be able to compare these different motifs, or the different k-mers in terms of how often they occur. Right? So one of the issues is, if you have a neuron, if you have a neuron that has a lot of these C nodes, right? So the axons have more C nodes, a lot of them, but some of them have fewer, right? An axon with a lot more C nodes than another is also going to naturally have a lot more CC dimers, even AC, CT dimers, all of those, right? You would expect there to be more because there's more of the C nodes. So we wanted to normalize for that, right? If we're looking at dimers, or when we're looking at trimers, we want to control by dimers. The whole idea is to look at information at that size above and beyond what we already know. So to do that, we generate virtual, what we call surrogates, that are constrained by the level below. So if we're looking at dimers, we constrain by the length and the percentage of C nodes. And then we make a distribution for any given dimer type. So the AC and the AT we are having as an example here. We count those up, and we make a distribution. And then we see where does the count for this one fall, right? So how similar is it to the baseline, or does it fall well above, right, to say that it has more than you would expect, right? So in this case, the AT happens a lot more often than you would expect in this one, but the AC, just a little bit less than you would expect, right, at 22nd percentile versus 98th. And we use 100 surrogates per sequence per K. And this process is something where there is a process where you can just sample from uniformly from the distribution of all topologies for a given size. One of the problems that came up, though, is there are a lot of shapes of tree topologies. Right? You can see with the number of bifurcations, this goes up exponentially. And if you have to just generate those and only take the ones that fit your constraints, you have to generate a lot of trees, trillions of trees, if not orders of magnitude even more than that in some cases. And that takes too long. So for dimer constrained and above, I produced an algorithm that uses the dimers themselves, uses the trimers, and assembles the tree. And there was a little bit of an artifact ultimately in terms of a deviation from what we want to do with uniform sampling. But I was able to minimize that to the point where for trimers, it was for 2 of the trimers were only off by 0.2%, and the rest of them less than 1/10 of that. Right? So we have a distribution that's really close to our target distribution for a baseline. So I'd said before, where do we stop this analysis? Right? We want to find something useful and interesting, but we don't want to go beyond that. So here we're looking at the proportion of neurons whereby their percent C is fully captured. Is not fully captured, right? So ultimately, when this gets down to 0, we know that our work is done, right? At this point, there's about 13% deviation. There's information here that's worthwhile to us. If we go to dimers, there's still a lot of information that's worthwhile to us, right? We've only gone down a little bit. We go to trimers, and we drop down quite a bit. We're at about just under 2%, right? So there's definitely information there, but we can expect there to be substantially less informative information than before. And once we go to tetramers and pentamers, yes, there's a little bit of information, but it's not worth going into that. Perhaps in some subset of the data, we may find that there's some interesting information here, but in this analysis, we stopped with the monomers through trimers. So this is a first look at the dimers. And one of the things, so before when we looked at those C nodes, we saw that the axons and dendrites were pretty substantially different. They're functionally very different, and we would expect that the dimers perhaps would also be substantially different. But this is where we got an interesting surprise. The dimers are actually very similar between axons and dendrites. Apical dendrites, which were in the middle of the two before, are actually more different from the other two and strike pretty close to the baseline. So naturally, the terminal growth and segmental growth that we had looked at before came up as a possible explanation. So if we just look at the axons and the dendrites, not the apical dendrites, right, we get these. And we looked at a few, and this is a sort of a representative look at two of the different. So the CT is a motif, right? TT is an anti-motif, and we want to see why that is. And so we looked at terminal growth, and we saw that actually terminal growth explains a lot of it, right? So if you just take this and we grow it in all different ways, right? There's two different branches that we can branch off here, but they both result in this structure. There's two different ways, right? We can branch off of here or one of these two and get these two different structures, but there's two different ways to get this and one different way to get this, and so on. So we go until there's 4 bifurcations, and we see that CTs, there's 20 of them, and there's only 4 of the TTs. So that already gives us an indication of the sort of process that produces some of these motifs. Right? It's not the entire story, but it is, if we look at a number of these others, it definitely explains a large proportion of what's going on with these dimers. And so here's an example of an axon and dendrite from the mouse cortical and mouse cortical interneuron, and you can see those CTs, where they are, what they look like, and how frequently they occur compared to those TT dimers. Right? And this is another representation that's a little bit difficult to see, but the darker regions, again, are those motifs, CT motifs. So now if we go and look at trimers, right, again, there's less information here, and as it turns out, all the axons, the dendrites, and the apical dendrites were all a lot closer to that baseline. They didn't deviate as much, but where those were deviations, we saw again the axons and the apical and the dendrites largely looking like they're coming from terminal growth processes. Right? So we're seeing a recurring theme. But we do also see a few differences. These 5 trimers differ, and so you can see where the ACC is very popular in this dendrite, and the ACT is very popular in the axon. So why is that going on? One of the most likely features, if we look at TCT and ACT, that's basically saying the CT, you saw a bunch of those before, the CT motif, either on the larger side or the smaller side of any given bifurcation. We already know that axons are a lot larger, right? So you would expect, and it is the case, that those CTs are tending to fall on the small side, whereas the dendrites are a lot smaller, so they have more balanced. So that drives a lot of this. And that actually, when we looked at that distribution relative to that terminal growth process, that explained most of what was going on with the axons. But certainly we know there's got to be some deviation from that process from those %C, and indeed there was still a little bit left to discover. So we didn't dig down past that, but there's certainly a greater complexity yet to look at in terms of axonal growth and why they're structured the way they are. So in conclusion, motifs are those frequently occurring local patterns. Axons, dendrites, and apical dendrites differ a lot in those node type proportions, but axons and dendrites are a lot more similar when we're looking at dimers and trimers and so on. And a lot of these are really reflecting terminal growth, whereas the apical dendrites, I didn't show this, but they're much more reflecting a segmental growth process. But really, those axons, there's certainly more to look at there. There's more than just the terminal growth. But that is a bigger trend than I think we would have expected starting off. So the next thing that we wanted to do was do a global alignment. This is the idea here this is what comes from the sequence, the gene sequencing idea, right? You want to look for similarity between sequences, you match them up and see where they match and how well they match, right? And this allows us to relate neurons directly and then go on to look for clusters. We want to see where are they similar, and do those similarities reflect known information about the cell types and the brain regions in the species? Is there anything new that we find from that? And then ultimately to extract the regions of similarity and say, what is it in terms of these patterns that make these things different? Right? And we can relate that to functionality, to function. The process, we first started off just using a pure sequence alignment approach that's used in genetic analysis, and we didn't get any results any better than just using the sequence lengths, right? So the size of the tree was really all we got. We realized that we had to do something a bit different. And ultimately the issue was the normal sequence alignment doesn't take into account the treeness of the sequences, right? That there's a structure to them already inherent. So what we did was we made use of the idea of what happens when you add or remove branches. Right? So we'll start off with this tree, and if we were to remove this bifurcation, we end up removing a C and creating in the alignment what we'll call a gap. Excuse me. Now that was just removing a C. What happens if we remove one of the branches on a T node? Well, we end up losing that T, of course, and that A becomes a C. So we now have two different types of transitions: the insertion or deletion of a side branch, a C node, or the transition between an AT and a C. So from here to here. And then, of course, by that process, right, once we have gone from here to here, we can go one step further and get rid of the C. So you can have the case where either a C replaces an entire subtree, or that entire subtree can just disappear, as we see here. And that drives the alignment process, right? So here's an actual alignment between two sequences. And you can see the blue bars where they match up, and we have those gaps, or those orange X's. And so here are cases where we have an AT, right? It's a whole subtree that is missing in this other sequence. And in this case, all of these purple brackets show the case where you have one of these side branches, these C nodes, right? Or let's say this, matching up with an entire subtree. And those can get pretty big. And then we can count up the matches, count up those gaps, produce a score. But what we wanted to do also was to not allow the size differences to have too much of an influence, right? So if you have a small tree sequence matched up against a large one, it's going to more easily find ways to match the larger one than if you had a larger sequence against another large sequence. Okay, but one thing that you could say is, well, there's going to be obviously a size difference. We also wanted to minimize that size difference effect. And one of the reasons, again, going back to that issue of the axons, right, we know that axons are not always complete. So perhaps if we were to not do this, we might miss similarities between axons because of the great diversity in terms of size, because of how they're reconstructed. But if we normalize by size, then perhaps we can still capture the patterns, and that's what we really want. So in this case, we have one that's less than what you would expect from the baseline, which is just random trees of this length and this length, right? They're matched up against each other, you get scores, you get a distribution, and you just basically adjust this raw score and see where does it fall within the distribution of scores for random sequences of these lengths, right? So this is a little bit less than average. And so we can then look at actual ones. So I mentioned before about the different types of apical dendrite from the CA1 region of hippocampus and the CA3 region of hippocampus. And you can just look at them by eye and see some differences when you look at the dendrograms, right? So this is just pure topology ripping out all of the angles and the lengths and the diameters and such, right? And when we run the scores, we see that here, these two, when they're CA1, that's above average. These guys happen to have a different score, but different lengths as well, so they end up having the same above average score, right? Whereas all of the cross comparisons are either less than average or not very much above. And then we can look at the whole distribution of all of these possible comparisons and again see that matches, these examples match up pretty well with what we see here, right? When we compare between the classes, they're less than average. When we compare within the classes, they're better than average. And this suggests that we should be able to really distinguish different classes of neurons with this approach. So to do that distinguishing, we went and we built up a space, right? We had similarity measures. So we could just use similarities to do certain types of clustering, but there are potential issues with those sorts of clustering. We can do a lot more interesting analyses if we have a space to work with, right? So we built up a multidimensional abstract space where if you have two neurons that have, or two neurites that have a very similar score, they're going to be very close together in this space. We'll say this is one dimension, this is two dimensions, right? There could be multiple dimensions beyond that, but start looking at this. So imagine this is a density of all of the neurites in the space. So that means at this point here, there's a lot of neurons right in this central area. And there's fewer here, but you can still see that there's definitely a bulk, right? So this suggests that we have, you know, a whole bunch here, but some over here and some over here and so on, right? But we can let the computers figure that out. So we do what's called model-based clustering, where we have a basic idea of the sorts of shapes that these clusters could take. They're multivariate Gaussians. And then we try a whole bunch of different number and different parameters and end up optimizing for that. And once we get that optimized set of clusters, we can then look and compare with the known types. So if we start off with all the data, we'll break it down by the most obvious differences, the axons, apical dendrites, and dendrites. And we see that there's 4 dendrite clusters, 2 axon, and 1 apical dendrite cluster. And that is to say that there's more apical dendrites in this cluster than you would expect by chance, and there's many fewer in this of the dendrites and axons. And the same is true for the other cases. So that's pretty nice. We would hope to get this kind of result, and we also see that we have subclusters in there. But we know also that we have differences in the apical dendrites, right? So when we actually cluster these separately, fortunately, we do in fact get those distinctions that I had talked about before, right? Here's the CA3 and the CA1 hippocampal apical dendrites, and here's the neocortical set, which is obviously a lot bigger, as I had mentioned before. There's a lot more neocortical apical dendrites. Another thing we wanted to see was, is this space, does it provide more information than if we just took basic topological measures, that asymmetry measure I talked about, the number of branches, the coalescence, as well as the maximum branch order, right? How many branches from the root, the maximum number from the root to a tip. And so we did a classification, one using clustering and one using a trained classifier. And we found that in most cases, actually, they perform about equally, but there are a few cases in which this space actually performs better. Right? And so this is just showing that in one measure, this was, I'll just talk about the accuracy here, 94% accuracy in this case, which was a few points better. And there was another case in which it was about 8 points improvement over the topological metrics. So we know that we have something uniquely informative here, and we're not just reinventing the wheel. So now we wanted to look at more specific clusters, and I won't spend too much time here, but the idea here is basically to say what are the counts in the different classes, what are the counts in the different clusters, what are their intersections? Make a contingency matrix. I did a chi-squared test, which basically says, are there associations in this table? The answer was very clearly yes, and the next question was, what are those associations? So we looked to see, we basically get these residuals, and those can be turned into p-values, right? How likely is this to have happened by chance? And so I actually turned these took the log. First of all, I did a multiple testing test, because there's a whole bunch of different tests here. So we corrected for that with the Bonferroni correction, and then took the log, right? And so the higher the values or the lower the values, if they're very strongly negative or strongly positive, that means there's either a very strong positive association or a very strong negative association. And so here I've highlighted all of those positives and negatives, with the color being the positive and the dark being the negative. And what we end up seeing, obviously there's a lot of clusters I could go into, but I won't go into too much detail, but we see these 3 clusters correspond with 3 different cortical types of neurons. And these 3 clusters are associating with motor neuron and sensory neuron. So we certainly see a differentiation based on those types of, those distinctions between general classes of neurons in the dendrites.
Could you point to the clusters again?
Certainly. Up here? Or
no, there.
Here, okay, so yeah, so we have cluster 1, 2, 3, 4, 5, 6. The colors correspond here. I've highlighted different ones here because these are different spatial dimensions. These are the first 2 spatial dimensions of the space. These are the 3rd and 4th. It requires that to really get the separation. And one thing to bear in mind when we look later is we have a lot more dendrites than we have axons, right? And also more types, right? So we would expect to get more clusters, and indeed we do. And we also expect to require more space to represent the differences, and we do. Right? And so this is just a look at some of the example cases. And you can see to some degree by looking at the dendrograms, the differences. And of course, one of the interesting things is here, in terms of other morphological features, they actually may be fairly different. But the topology is still something that we're interested in distinguishing from those other features. And we see, for instance, these 2 purple guys, right? These are 2 different types of sensory neurons. And overall, they definitely look pretty different, but their topologies are very similar. So certainly be interested to understand why that is. But the next thing that we're going to really spend a little bit more time focusing on is the axons. Right? We had fewer clusters, we had fewer total axons, we had fewer types of axons, but we did still find some very interesting results. So one thing is the Drosophila olfactory bulb, the sensory-related neurons, take their own cluster over to the side. You see an example here. And they didn't overlap too much. Now, one of the other things that we saw, we have different types of pyramidal cells from the neocortex, but one of the biggest distinctions we ended up finding was these ones are from the rat, and these ones are from the mouse.
Huh.
And we didn't necessarily expect beforehand that we would see a difference just between mouse and rat. They're two different species, sure, but they're both rodents, and we know that they're similar in a lot of ways. So one of the thoughts that we had that might explain it is, well, they're definitely from different archives. So these rat ones are from from 3 different archives, and these mouse ones are from one specific archive. So perhaps there's, and also obviously there's the possibility that you have an effect of brain size, right? The neurons are bigger because the brains are bigger. So perhaps for rat, you're getting some cut off more, like I had mentioned before. But in addition to the rationale for doing that whole normalization process, so that if they're different size or something gets cut off, hopefully they still look the same. And I did do some testing to see if we actually manually clipped off a bunch, when they're above or below at a certain distance in space, what does that do? And we found out that those normalized scores, comparing one neuron to itself when it's been clipped off, actually are still really high. So the chances of the brain size being responsible for this difference, we haven't ruled them out, but it definitely seems unlikely for that to be the case. So there still could potentially be other lab, reconstruction method artifacts, but it could actually also be that there really is some difference in topology between the rat and the mouse cases. So future exploration of that will tell. But the other big interesting thing that we found, so interneurons are known to really have various different morphologies, and they certainly have a number of different roles. And I'll discuss in a bit what might be going on here. But we found out that the dendritic targeting interneurons tended to fall actually in this mouse pyramidal cell cluster, as opposed to the perisomatic targeting interneurons. And so we decided to take a look, as I mentioned before, what are the similarities, what are the differences? So to figure that out and extract those similarities, we did a multiple sequence alignment. It's a little bit difficult to see, but basically every line here is a sequence from a cluster. And if you look vertically, you can see places where they really tend to align tightly. Right? We zoom in, we see, again, a tight alignment here, here, and so on. And if we threshold only those positions whereby a lot of the sequences, at least 50% of the sequences agree, we call that the consensus. And then we can put that all together. And so here's the consensus for that third cluster of the pyramidal cells and those dendritic targeting interneurons. And basically this is shows the consensus dendrogram, sort of what that structurally looks like. And the greater the width of these means they're more highly conserved. And then we can compare that. So these are example cases, which unfortunately are not easy to see on this screen. I apologize. But we can compare that to the fourth cluster, which we talked about, and very clear night and day difference, right? This is a lot more symmetric than this, and there are potentially other features to dig into and look at, but even just looking at the symmetry differences, this sort of brought up 2 possibilities that I had considered with Giorgio. And one of them is something I had talked about earlier in terms of the path length management, or right, you want to minimize the amount of path length, or do you want to minimize the total amount of wiring? Right? And one of the reasons you might minimize path length would be to make it so that the timing, right, when a signal leaves the soma and gets to its target, might be more similar. We know that these interneurons, the perisomatic targeting ones, mostly basket cells, we know that they're involved in synchrony, synchronizing brain regions, keeping a brain region operating at a particular rhythm, right? Those rhythms that are in your brain from an EEG or what have you, those are driven by interneurons cycling, basically. So possibly they need to have a tighter timing schedule, right? That their signal needs to reach their point at the same time or closer in time. That's one possibility, right? And if that's the case, then we get a more symmetric arbor. Another possibility might actually be, where are they targeting, right? If they're trying to be synchronous, they might be targeting a very dense local region, as opposed to others that are doing things like pattern separation, reaching out to a greater diversity of neurons, which we know for pyramidal cells are the principal neurons, right? That they need to do that in order to have a complexity in a network. So perhaps those dendritic targeting interneurons also benefit, or the network may benefit from them having that sort of a diversity in their targeting. And that may be the relationship. And they're not mutually exclusive, those both could be the case. Now, we did do a little bit of analysis to test that, and In terms of the spatial distributions, it looks like spatially they're reasonably distributed the same in terms of distances, but where those fall requires a bit further analysis to see. The lengths, though, are definitely different. So that suggests that we might indeed have a higher local total amount, total density for those cells. So one other thing we wanted to look at to bring the consensuses and the motifs together is, do they ultimately give the same information, or are we getting something unique from each of them? So we look to see the consensuses. We actually do a motif analysis on the consensuses and compare those to the motifs of their host clusters. Do they match up? And we actually found that only 14% of the variance in the motifs of those clusters is captured by the consensuses. So there is certainly some relationship there, but we're not catching much, and they're definitely seeing something different. Another way to look at it is these are the motif profiles of clusters 1, 3, and 4. And you can see that cluster 1 and 4, the green and red, are pretty similar. There are some differences, but they're pretty similar, especially compared to this blue cluster 3. And then you look at the consensuses. You see cluster 1 and cluster 4 are very different, and cluster 3 sort of falls somewhere in between. So again, it's starting to look like the global structure topologies that we get out of the consensus process gives us something unique from those local motif values. And then finally, we also took those surrogates that I talked about earlier, right? These are random trees, but they're constrained by those motifs, and built consensuses from them. Right? We did the alignment consensus process from them. And there are some cases where actually they match up nicely, some of the clusters, but there's also some, like this case, where clearly they're very different. Right? They still do better than if you just used length-constrained surrogates, but all of these together show us that consensuses and motifs are giving us complementary topological information. So practically speaking, in my dissertation, I've created the diadem metric for enabling reconstruction, improvements in the automated reconstruction process in the field, created a neurite sequencing procedure, applied that to motif analysis, which has shown us some interesting information, a multiple sequence alignment and consensus extraction, and also the global pairwise alignment. And one of the interesting things that this can do, aside from the analysis that I did, is we can also use it to detect duplicates. Right? Curation of a database is important. If at some point you have two neurons that both come in that happen to be the same but different reconstructions, you want to be able to detect that. And so we actually did detect 19 duplicates in the process of just looking through the data. And so we were able to weed those out of the 10,000 neurons in the database at the time. And we'll be actually looking to insert that into the pipeline in the future. And we also show that we can detect duplicates that have edits made to them. And also this can be applied to search. And so the scoring procedure I used before can be modified. So we look at how many matches of a given score or better would we expect, the expectation value. We can sort of adjust the scoring method to give that, and that can tell us something about how similar, how related, right, are particular neurons. I want to see what are the ones that are like for this other neuron that I'm looking at, different species, does it match up well? The search is something that's very much used in genomic analysis. So when we have the big data comes, that can be very useful. So in the future, we really would like to look at an expanded representation with branching angles, with tortuosity, and really whatever someone might be interested in. In the future, it would even be great if we could have measures of, say, spine densities or particular protein densities. Those sorts of things could also be really useful. And by doing that, we could investigate branch-level multi-feature patterns. We'll also have greater specificity, right? We only have 3 characters right now, so the score ranges are going to be compressed to some extent by that. We can have a much larger score range and perhaps potentially see a lot more distinctions if we have more data in the representation. It's also the case that I didn't really optimize this, but this process is very easy to optimize in a whole bunch of different ways. I won't go into that unless anyone has questions on that later. And there's also that we can easily create a local alignment process, which maybe could find domains, especially when we have really more complete axons. There might be subregions that have some specific architecture to them that some sort of a local alignment might give us. So just to wrap up, axons and dendrites, I showed, are topologically more alike than we would first think. Most of the topological the motifs that we found are at least primarily driven by growth mechanisms. The sequence representations of neuronal morphology capture at least as much information, and in some cases certainly more, than our standard topological metrics. We showed that multiple alignment of related neurons allows us to identify certain consensus structural features. And finally, that the motif analysis and the consensus analysis provided really complementary type of information. So that concludes my talk, and I'd very much like to thank a number of people. First of all, my advisor, Dr. George Raskley, who has guided me wonderfully in my process here, as well as obviously on all of this work, and also has provided really great opportunities to do very interesting things. It's hard to see this background, but there was another very interesting art project, which you can see downstairs later if you'd like, that we worked on together. That was really great. Just Dr. Jim Olds, who I very much appreciate from helping out early on, starting the program, the classes, learning about how to write grants and what academia life is really like, and a lot of interesting experiences there. Dr. Josef Weissman, who came on about midway in my process and at a actually very helpful time to help me work through some of the ideas for the multiple alignment, which I think is maybe one of the more novel components of what I've done, allowing this consensus approach for tree sequences. And so there were some early insights and various things to try, and I very much appreciated that. John Grafenstedt and Ted Dumas used to be on committee. They both had to leave for different reasons. They've both helped me out in different ways. John Grafenstedt with bioinformatics, Ted Dumas with the wet lab stuff and learning how to record and what that is really like, and those procedures. And I'd also like to highlight Carrie Brown, who I've worked with on a number of projects and has been of great help, and a whole bunch of other people from the CN3 lab who I owe for advice, feedback, just ideas and talking and everything. And of course, the funding from NIH. You can't do science without some money to back you up. So thank you very much, and with that, please, if you have any questions. Thank you.
So how do you see with this, your consensus work, how do you see with the new big neuron project coming up to make it useful or work?
Right. So, I mean, the concept, I think, transfers pretty well, right? So the issue, so big neuron is a process to try to basically take multiple automated reconstruction results and put them together, and that requires a consensus sort of approach. We can certainly take these reconstructions. One of the differences, obviously, is big neuron, they'll want to join all of the other information, not just topology, right? But there are other processes to, say, take multiple paths and create a consensus path. So really, if we were able to combine that with the topological consensus process that we used here, that really should be, in relative terms, an easy thing to do. It's certainly going to take some development and whatnot, but all of the components are there, and I think that they will translate over pretty well.
Todd, the difference between the rat and the mouse topologies, is there any other possible explanation, like somatosensory versus visual region,
Right.
or primary versus higher level processing?
So looking by cortical subregion, and we did have a fair amount of information there, there didn't seem to be much of a signal. That doesn't mean that there couldn't ultimately be, but by layer, there definitely could be. So we definitely did have some layer information, but the layer information is probably, among the metadata that's sort of least complete. So that could well be the case. You could have layers that, projecting from, I guess that's the other thing, right? So even within a layer, you can have a neuron that projects to one region versus another. And so you can certainly have diversity there. So yeah, so it absolutely could be that one of the labs maybe ended up capturing a certain type of pyramidal cell from different layers, or projecting to different postsynaptic brain regions. But we'll definitely have to look as more data becomes available, especially on the metadata front.
So maybe you can comment a little bit about it. If I'm not mistaken, the way that you try to look for associations between the clusters and the metadata was to take the metadata in the main dimensions that come from species and brain regions and neuron type,
right?
And look manually, so to speak, or all-to-all for possible associations between the emergent clusters and these metadata.
Right.
It strikes me that in the ideal world, you'd be double blind to both dimensions, so you'd feed in all the metadata that are available, and ideally that would be all the metadata, period. Like all the metadata would be available.
Right.
And then you would let a computer not only just find the cluster, but also find the best association. So if it says, hey, the best explanation is given by the objective type, whether it's dry or oil immersion, so be it. Or it might be the laboratory of origin, whether it's in vitro or in vivo, and so forth.
Mm-hmm. Right. So certainly, first of all, obviously subclustering, because I think to some degree, when the differences between certain brain regions or species are sufficiently large, I think we can sort of segregate those out. So if we were to do this, for instance, we could get rid of those olfactory neurons from the fly. I think we know those would be pretty different. But then we could keep, say, all of the pyramidal cells and maybe even the interneurons and take that approach. And one of the challenges comes, though, is that in terms of to capture some of those, you may need a larger number of clusters, or you may need to drill down and focus on a smaller subset. So it's not just a matter of feeding in all of the potential metadata. It may also be a matter of different combinations of clusters and subclustering to find that. And with a large enough data set, especially if we do continue to really grow as it we clearly are doing in terms of the amount of data, we may be able to actually make some detections. I mean, even now, obviously, those p-values were really, really small, so that suggests that maybe we could find those things. But obviously, the more tests we do, the greater likelihood of false positives, and also the more metatypes that we feed in, the smaller numbers that they have, and so the more difficult it is to detect an actual association there. But nonetheless, there's certainly some of those that we could look at by picking those metadata features, that any one that has a sufficient number in them, and we could really just throw that all. And we just need to come up with a process for doing all of the different comparisons. But that would definitely be a possibility, and could produce some additional results from what we saw.
Todd, your performance for classifiers is somewhat better in the alignment space compared to the topological space. So is this difference in performance statistically significant?
At least in one of the cases, I'm certain it is. And I believe in two of them. I have not done the complete statistical analysis to determine exactly what the confidence intervals or the statistical significance is for those. But like I said, for the one of them, just looking at the difference in percent, I'm certain it is based on multiple runs. But one thing that we also would really need to do is do more larger data sets with more data to test. But I did, at least for all of them that I could, did the classification, right, the training with one set, and then brought in another set to do the testing. And so, that gave me a lot of confidence for at least one of those. All of the rest of them were right within the percentage, which is, I'm sure, given the diversity I saw even between different runs, right? So the linear discriminant analysis is stochastic. It will vary from one run to another. And so I did a bunch of those. And at least in terms of that approach, I did see statistical significance. I didn't actually look for the ones that were in 1% because that just, seemed most likely just chance.
So
correlation between the dendrite and axon sequence of the same neuron? So if you
So, I mean, if you were to just look at one and another and compare them, they're certainly very different. But if you're talking about how do you see differences, if you were to compare two different neurons and take a look at the axon and the dendrite of each, would you get sort of clusters that would segregate out in both cases? I didn't specifically look at that, but just from the results that we saw, it would suggest that in most cases you probably would. But again, with the axons, just given the diversity of the data, it's a little bit tricky to tell. But I would imagine yes, and certainly in some of the cases we did see that. For those cases, we did have the data available. Yeah.
So we're going to continue asking questions to Todd in closed session from the committee. So if there are any last questions or curiosities or debate from the public, this is a public talk. So this is the last chance before we close doors for a little bit.
All right. Well, thank you all. Feel free to grab a little bit of food. Excuse me, on your way out.